myeloma is the second most prevalent blood cancer, represent 2% of all cancer patients. And when I came into this disease in the 90s, patient would be lucky if they would make it 2, 3 years. And the standard of care were melphalan and transplant, which was uh, invented in the 1960s. So there was a first revolution in the early 2000s when proteasome inhibitors and IMiDs came along. And then in the middle of the second decade of the 21st century, when new immune drugs, anti-CD38s and now CAR-Ts and bispecific came along, and we are finally starting to see some deep, immune responses, deep, durable responses in our patients. This being said, there are still about 1/4 of the patients who are not benefiting from these innovations, these new treatment. So that's the first issue. Second, when patients start relapsing, um, these relapses are really not very good, especially after these new immune agents that we've brought to the clinic. So that's one big problem. The other big problem is there are precursor conditions in myeloma called monoclonal gammopathy of unknown significance, MGUS, or smoldering. And the standard of care in myeloma is still observation. And that doesn't make much sense because for most, if you have a skin lesion, if you have a colon polyp, you remove those. You don't wait for the disease to metastasize everywhere. That's kind of what we're doing with myeloma. The issue is the predictors that we have right now are barely more reliable than flipping a coin. This is why clinicians hesitate to treat. And the last issue is, even with these, new immune agents, BCMA is a great new target for myeloma. You have bispecific CAR-Ts, drug conjugates. We already are in this post-BCMA era. We need to look for what are the next new immune drug we can bring along and combine with these other agents. Combination is what has made the difference for other cancers. And that's where we need to get going. So this is where the MMRF come into play. We've always been funded by a myeloma patient, Kathy Giusti, who, when she was diagnosed in 1998, was told that her then 1-year-old daughter, she wouldn't see her to get into kindergarten. But given her industry background, she said, I don't think so. Founded this foundation, and very early on, we realized that we need to go beyond just funding research. We need to be deeply involved in the research. So we were really the first true research nonprofit foundation. So we created back in 2004 a clinical consortium, which now has about 24 centers, the who's who's of multiple myeloma, the MD, the Dana-Farber, Mayo Clinic, MSKCC, and everyone. And through this clinical consortium, we've every single drug approved in multiple myeloma, came to this consortium one way or another. We ran 90 clinical trials. And again, this is how now we have our patients who can make it 7, 8, 9 years. But a lot of these drugs, all of these drugs are going after what we call the plasma cell biology of the disease, the fact that they are plasma cells. So they express at their surface specific molecules that you can target because plasma cells make these antibodies. They are a factory to make proteins, and anything you can throw at them that disrupt the protein machinery, these cells won't like very much. This is how proteasome inhibitor works and IMiDs work. It became apparent that we needed to know more about the blueprint of the disease, which was a big black hole. So we started a consortium back in 2004 to map the myeloma genome. And that happened when we were sitting in Bethesda right next to Eric Lander, and he was presented his view of what the Cancer Genome Atlas, the TCGA, would be. Myeloma was not one of those, and we said that's unacceptable. How can we change this? And Eric said, well, we need a few hundred samples, a couple millions, and we'll make this happen. And we did. Turned out in 2009, of all the cancers, multiple myeloma was the first one to sequence a large number of its genome. And it was a great effort, but it was a mix of newly diagnosed and relapsed patient, one snapshot in time. And we said we need to do much better than this. We need to follow a large number of patients from their very first diagnosis all the way throughout their journey with disease. And that became our COMPASS study. And through this effort, we are following now almost 1,200 patients doing deep genomic and immune analyses on these patients, looking at how the disease changes to outsmart the treatment we throw at it. And that was a wonderful effort, generated over 100 publications, will be remembered as one of these seminal clinical genomic efforts in any cancer. For a long time, it had the biggest amount of genomic data out there in the public domain one could use to derive hypotheses. And through this COMPASS effort, we discovered, among other things, that almost 3/4 of myeloma patients had the so-called actionable alterations, alterations for which there are drugs out there in other cancers that could be redeployed to myeloma. And that became our MyDrug master protocol. For example, drug like MEK inhibitors, which are used in skin cancer, melanoma, to go after an alteration, BRAF, found in 60% of melanoma patients, is found in about 8% of myeloma. So through these kinds of approaches that we have learned through COMPASS to unlock and decipher the genomic blueprint of the disease, we can basically look at the specific genomic background of a patient, figure out if there are Achilles heel in there that we can target from a drug perspective and go after them. That's called, in some way, precision medicine, and that's something we've been doing since 2004. And now we're on this deep genomic knowledge, we're adding also these new technologies, deep immune profiling of these patient samples. So COMPASS was great, 1,200 patients, massive amount of data, a lot of publications. But what we realized is even 1,200 patients was not quite enough in order to make precision medicine a reality for all of our patients. So this is where this follow-up effort called CureCloud was initiated. We said we need to marry deep science with population level real world evidence. How can we bring those together in an efficient way so that we can learn what's going on in each and every patient and figure out what's the best treatment for his or own specific form of the disease. CureCloud's an interesting effort. It's a direct-to-patient initiative. Basically, individuals can just go on their smartphone, they can consent, and what that does is it bring a mobile phlebotomist right to their home, take blood, and we can do deep genomic and immune analyses on that blood to figure out to do some research and figure out if there are some of these actionable alterations. It's bringing the power of genomics to the masses. One of the big findings of COMPASS also was that these diagnostic tools that we had since the 1960s or 70s cannot really accurately predict well the disease given what we know today. You need these next generation technologies in order to do that. And it's not available in, if you go in one of these big cancer centers, you will get these tests. If you're in a small countryside clinic, you may not get that. So it's one of our goals to bring this power of these new technologies to all patients, again, to help them, with their doctors to navigate their disease. So Cure Cloud is ongoing. We have enrolled at this point almost 1,000 patients. Started last summer. We're basically enrolling 50 to 70 patients every month. And we're starting to learn quite a bit about that. We made some presentation at the last scientific meeting. We're going to make more at the upcoming meetings this fall. Cure Cloud is also interesting because it can help us address other big questions out there. Through Cure Cloud, we are looking at this issue of precursor condition and can we find better predictor and better approaches. We're looking at what's going on in the real world with these drugs that all these patients are receiving. Who are those who are responding well? Who are those who are responding poorly? Can we find better approaches for our patients? New issues that have emerged. One obviously is COVID, and this COVID pandemic has dramatically changed the way we do cancer drug discovery and cancer treatment in this day and age. Think about telemedicine. Think about you know the way that we were not able to bring our patients to the clinic. So we basically would have to find regimens, either oral drugs or like short infusion that could do the trick for those patients. Obviously, COVID taught us that disparity, which, myeloma was always a poster child for disparity. African Americans get multiple myeloma at 3 times the rate of people of European descent, and they get it typically 10 years younger. So that was always a problem, but disparity has magnified this, COVID has magnified this issue of disparity. And COVID, now what we're seeing in our patient through, again, our clinical consortium is that up to 1/4 of myeloma patients get low or zero serological immune response to vaccination. And that's a big problem because we obviously cannot keep our patient in lockdown forever. So we're involved in an effort right now, bringing the power of Cure Cloud, that of another large COVID effort called Impact, which is done by one of our collaborators at Dana, to do this deep immune profiling that we've learned to do to our various efforts and figure out what's going on in those patients. We know that the immune response is a mix of B cell. The B cells are the one making the antibodies, and the T cells, the one that kind of help these B cells along the way. So it may still be possible that there is some T cell response if there's no antibody. We doubt that's the case because some of the recent experience with these recent effort doesn't seem to tell us that that's the case. But so we're really trying to figure out what is deficient. Myeloma has this uncanny ability to paralyze and evade the immune system. Myeloma is a cancer in some way of the immune system. It has evolved mechanism to tell to the immune system, hey, don't attack me, I'm one of yours. And it could be the case here that that's what we're seeing. So we're trying to figure out what we can do for our patients, and this is where Cure Cloud is great. And again, the other piece about Cure Cloud is we want to bring this solution to each and every patient, wherever they are in the country, whatever the color of their skin, whatever their social situation. We want to level the playing field for everyone. So, and again, we're going to have a big disparity effort that we're going to attach to Cure Cloud later on this fall, and you will hear more about that one.